Structural polysaccharides. These polysaccharides make the structural framework of the cell wall in plants and skeleton of some animals. These are of two types, that is chitin and cellulose. Chitin is the second most abundant organic substance. It is found as the structural component of the fungal cell walls and exoskeleton of arthropods. It provides both strength and elasticity to the structure it forms in chitin monomer. Unit is a nitrogen containing glucose derivative known as the N-acetylglucosamine. It has an unbranched configuration and monomers are joined together by 1 to 4 beta linkages. Cellulose is a fibrous, tough, water-insoluble substance found in cell wall of plants. Some protest paper made from plant pulp and cotton fiber is cellulosic. Cellulose is a homopolysaccharide composed of beta-D-glucose. Units linked with the beta 1 to 4 glycosidic bond. Cellulose molecules have unbranched and linear chains. Cellulose does not contain complex helices and hence cannot hold I2. No complex helices. And can't hold I2. Then we have the mucosubstances. Some heteropolysaccharides serve as mucosubstances. These mucus or slime forming substances, they are of two types. Mucopolysaccharides, they are slimy substances which possess acidic or the animated polysaccharides formed from the galactose. Mucopolysaccharides, these are the slimy substances which possesses acidic or animated polysaccharides formed from the galactose mannose. Slimy substances, sugar derivatives, and the uronic acid. Mucopolysaccharides are found in both plants and animals. They may be attached to a protein molecule to form a proteoglycan. Proteoglycans provide the ground or packing substance of connective tissue. Example: hyaluronic acid, chondroitin, sulfate, and heparin. Mucoproteins or the glycoproteins are the next. The protein with conjugated mucosaccharides form mucus. Conjugated mucosaccharides forms mucus. These are found in the stomach, nasal secretion, intestine, vagina, and are antibacterial and protective in function. Then we have nucleic acids. These are polynucleotides together with polysaccharides and polypeptides. These comprise the true macromolecular fraction of any living tissue or cell. These are polynucleotides. For nucleic acid, the building block is the nucleotide. Nucleotide is the building block. A nucleotide has three chemical distinct components. One is heterocyclic compound. The second is monosaccharide and the third is phosphoric acid. Phosphoric acid or phosphate. As you notice in the heterocyclic compounds in nucleic acid are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. Adenine and guanine are substituted purines. While the rest are substituted pyrimidines. The skeletal heterocycle ring is called as purine and pyrimidine respectively. The sugar found in polynucleotides is either ribose or 2-deoxyribose. A nucleic acid containing deoxyribose is called deoxyribonucleic acid. Deoxyribonucleic acid is DNA. While that which contain ribose is called the ribonucleic acid. A nucleic acid containing deoxyribose. In the structure of proteins. Being macromolecules, proteins are not freely soluble in water but form colloidal complex with the same. They form colloidal complex. Chemically, a protein is made of carbon, hydrogen, oxygen, nitrogen, and sulfur. Some proteins additionally contain phosphorus, iron, and other elements. Every protein has a three-dimensional structure that corresponds to its function. The four-level structural organization in protein are primary, secondary, tertiary, and quaternary. Primary is the linear sequence of amino acid in a polypeptide chain. Primary structure describes the basic structure of a protein, that is number and sequence of amino acids in each polypeptide. We have primary. It is a linear sequence of amino acids. It describes the basic structure of a protein. The structure of protein is most unstable. Next, we have the secondary structure. It is a development of the new steric relationship of amino acids present in the linear sequence inside the polypeptides. They are insoluble in water and fibrous in appearance. There are two types of secondary structure, that is the alpha helix and the beta helix. It is a right-handed rotation of spirally coiled chain with approximately 3 half amino acids in each turn. This structure has intramolecular hydrogen bond. It is between two amino acids of same chain. Example: keratin, myosin, topo, myosin. Beta helix has zigzag structure. Two or more proteins are held together by intermolecular hydrogen bond, that is fibroin, which is silk. Then we have tertiary. It is formed by the further complex formation, bending and folding of It is stabilized by several types of bond. It is peptide, hydrogen, ionic, disulfide, hydrophobic, van der Waals. This structure imparts a 3D conformation to the protein. Then the quaternary structure. It is a multimeric protein and consists of two or more polypeptide chains. Each polypeptide develops its own tertiary structure and function as subunit of protein. Example: insulin, collagen, hemoglobin. Keratin is a fibrous stuff resistant to digestion. Scalar protein hardness of keratin is due to abundance of cysteine amino acid in its structure. Peptide bonds are the strongest bonds in proteins. Majority of protein and enzyme in protoplasm exhibit tertiary structure. Quaternary structure is most stable structure of protein. Nature of bond linking monomers in a polymer. In a polypeptide or a protein, amino acids are linked by a peptide bond which is formed when the carboxyl group of one amino acid reacts with the amino group of the next amino acid with the elimination of a water moiety. The process is called the dehydration. The nature of bond linking monomers in a polymer. So amino acids are linked by peptide bond. In a polysaccharide, the individual monosaccharides are linked by a glycosidic bond. This bond is also formed by dehydration. This bond is formed between two carbon atoms of two adjacent monosaccharides and the nucleic acid and a phosphor moiety links the three carbon of one sugar of one nucleotide to the 5' carbon of the sugar of the succeeding nucleotide. The bond between the phosphate and hydroxyl group of sugar is an ester bond. As there is one such ester bond on either side, it is called the phosphodiester bond. Nucleic acids exhibit a wide variety of secondary structure. For example, one of the secondary structure exhibited by the DNA is the famous Watson-Crick model. This model says that DNA exists as double helix. The two strands of polynucleotides are antiparallel, that is, run in the opposite direction. The backbone is formed by the sugar-phosphate-sugar chain. The nitrogen bases are projected more or less perpendicular to this backbone but face inside. A and G of one strand compulsorily base pairs with G and C respectively on the other strand. There are two hydrogen bonds between A and T and three hydrogen. Two hydrogen bonds between A and T and three hydrogen bonds between G and C. Each strand appears like a helical staircase. Each step of ascent is represented by a pair of bases. At each step of ascent, the strand turns 36 degrees. One full turn of helical strand would involve 10 steps of the 10 base pair. Attempt drawing a line diagram. The pitch would be 34 Armstrong. The rise per base pair would be, this is a pitch, would be 3.4 Armstrong. This is the rise per base pair. This form of DNA with the above mentioned silent feature is called the BDNA. The above information. That is next is the dynamic state of body constituents. One of the greatest discovery ever made is the observation that all the biomolecules have a turnover. This means that they are constantly being changed into some other biomolecules and also made from some other biomolecules. This breaking and making is through chemical reactions constantly occurring in the living organisms. Together, all these chemical reactions are called the metabolism. Each of the metabolic reactions results in the transformation of biomolecules. Majority of these metabolic reactions do not occur in isolation but are always linked to some other reaction. In other words, metabolites are converted into each other in a series of linked reactions called the metabolic pathways. These pathways are either linear or circular. These pathways crisscross each other. This metabolite flow is called the dynamic state of body constituents. What is most important is that this interlinked metabolic traffic is very smooth and without a single reported mishap of healthy conditions. Another feature of these metabolic reactions is that every chemical reaction is a catalyzed reaction. There is no uncatalyzed metabolic conversion in living systems. Even CO2 dissolving in water, a physical process, is a catalyzed reaction in living system. The catalysts which hasten the rate of a given metabolic conversion are also protein. These proteins with catalytic power are named the enzymes. Metabolic basis for living. Metabolic pathways can lead to a more complex structure from a simpler structure. For example, acetic acid becomes cholesterol or lead to a simple structure from a complex structure. For example, glucose becomes lactic acid in our skeletal muscle. The former cases are called the biosynthetic pathways or anabolic pathways. The latter constitute degradation and hence called the catabolic pathways. Anabolic pathways are expected to consume energy. Assembly of a protein from amino acids require energy input. On the other hand, catabolic pathways lead to a release of energy. For example, when glucose is degraded to lactic acid in our skeletal muscle, energy is liberated. This metabolic pathway from glucose to lactic acid, which occurs in 10 metabolic steps, is called the glycolysis. Living organisms have learned to trap this energy liberated during degradation and store it in the form of a chemical bonds as and when needed. This bond energy is utilized for the biosynthetic, osmotic, and the mechanical work that we perform. The most important form of energy currency in living systems is a bond energy in a chemical called the ATP, which is the adenosine triphosphate. The living state. Tens and thousands of the chemical compounds in a living organism, otherwise called the metabolites or biomolecules, are present at the concentration characteristics of each of them. For example, the blood concentration of glucose in a normal healthy individual is blood concentration is 4.2 millimole per liter to 6.1, while that of hormones would be in nanograms per milliliter. The most important fact of biological systems is that all living organisms exist in the steady state characterized by the concentration of each of these biomolecules. These biomolecules are in a metabolic flux and the chemical or physical process moves spontaneously to equilibrium. The steady state is a non-equilibrium state. One should remember from physics that systems at equilibrium cannot perform work. As living organisms work continuously, they can't afford to reach equilibrium. Hence, the living state is a non equilibrium steady state to be able to perform work. Living state is a non equilibrium steady state to be able to perform work. Living process is a constant effort to prevent falling into equilibrium. This is achieved by energy input. Metabolism provides a mechanism for the production of energy. Hence, the living state and metabolism are synonymous. Without metabolism, there cannot be a living state. Enzymes are organic catalysts which catalyze biochemical reactions without being utilizing themselves. They occur in the colloidal state and often produce an inactive form called the proenzymes or zymogens. These are converted to active enzymes in the presence of specific factor like pH, substrate, etc., with few exceptions. All known enzymes are proteins, but all proteins are not enzymes. Enzymes are protein, but all proteins are not enzymes. Ribozyme, ribonuclease P, and peptidal transferase are three non-protein enzymes. Ribonuclease P, peptidal transferase. These are all the non-protein enzymes. Some nucleic acids behave like enzymes called the ribozymes. We have classification and nomenclature of enzymes. According to modern system of enzyme classification introduced by the International Union of Biochemistry and Molecular Biology IUBMB in 1961, enzymes are classified into six classes based on the type of reaction catalyzed. First is the oxidoreductases or the dehydrogenases. Oxidoreductases or dehydrogenases. These enzymes catalyze the oxidation reduction reactions by transfer of electrons. These enzymes are of three types: oxidase, example cytochrome oxidase; dehydrogenases, that is succinate dehydrogenases; and the reductases, that is nitrate reductases. Oxidases, dehydrogenases, reductases. Oxidase, we have cytochrome oxidase, D are the succinate dehydrogenase. Reductase are the nitrate reductase. Then next is the transferases. These enzymes catalyze the transfer of specific group G other than the hydrogen from one substrate to the another. The group transfer does not occur in free state. It transfer of specific group other than hydrogen from one substrate. Dash G plus S dash to S plus S dash. Then hydrolases. These enzymes catalyze splitting of larger molecules into smaller ones by addition of water, that is with the help of hydrogen and the hydroxyl groups. Usually bonds formed by dehydration are ester, ether, glycosidic, halide or PN bonds are catalyzed by these enzymes. Example amylase, sucrase, lactase. Then we have lysases. These enzymes catalyze the breakdown of specific covalent bonds and removal of groups without hydrolysis, producing double bonds or removal of double bonds by adding groups. Then we have isomerases. These enzymes catalyze the change of a Change of a substrate into a related isomeric form by rearrangement of molecules. These enzymes two are of three types: isomerases, epimerases, mutases. Isomerases change aldose to ketose group or vice versa. Epimerases change in position of one constituent or carbon group, and the mutases shift the position of side group like glucose 6 phosphate to the glucose 1 phosphate. And the ligases are that these enzymes catalyze the joining of two substrate molecules by utilizing the energy from hydrolysis of ATP, which catalyze the joining of carbon oxygen, carbon sulfate, carbon nitrogen, or phosphorus oxygen, etc. bonds. These are the joining of two substrate molecules by utilizing the energy from hydrolysis of ATP. We have characteristics of enzymes: proteinaceous nature. Enzymes are usually globular protein except two RNA enzymes. Efficiency. The efficiency of enzymes depend upon the number of active sites present over an enzyme. The number of substrate molecules changed per minute by an enzyme is called the turnover number. Turnover number is the number of substrate molecules changed per minute. Substrate molecules change per minute. The higher the turnover number, the more efficient an enzyme is. Higher turnover. Enzyme specificity. Enzymes are highly specific in their action. Different enzymes act on the same substrate but give rise to different products. Similarly, an enzyme may act on different substrates to produce different end products. Chain of reaction. Enzymatic reactions are not isolated, but they occur in quick succession, or a team of enzymes work together to perform multi-step reactions. Colloidal nature. Enzymes are hydrophilic and form hydrosol in free state. Reversibility. Almost all enzymatic reactions are reversible. However, reversibility is dependent upon energy requirements, availability of reactants, concentration of end products, and pH. Thermolabile. Enzymes are the heat sensitive or thermolabile and operate between 25 to 35 degrees Celsius. They become inactive at the freezing temperature and denature at 50 to 55 degrees Celsius and operate between 25 to 35 degrees Celsius. Enzyme substrate complex. The active sites of enzymes have crevices or conformations that fit in the substrates in the complementary fashion, forming a complex known as enzyme substrate complex. The latter are changed into product as soon as the products are released. The active sites become free to perform another catalytic act. Chemical reactions. Rate of physical or chemical process refers to the amount and product formed between per unit time. That is, rate equals to del P upon del T. Rate can also be called the velocity if the direction is specified. Rates of physical and the chemical processes are influenced by temperature among other factors. A general rule of thumb is that rate doubles or decreases by half for every 10 degrees Celsius change in either direction. Catalyzed reactions proceed at rates vastly higher than that of the uncatalyzed ones. When enzyme catalyzed reactions are observed, the rate would be vastly higher than the same but uncatalyzed reactions. For example, carbon dioxide plus water under carbonic anhydrase carbonic acid. H2CO3 is the carbonic acid. In the absence of any enzyme, this reaction is very slow, with about 200 molecules of H2CO3 being formed in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speeds dramatically with about 60 lakh molecules being formed every second. This enzyme has accelerated the reaction rate by about 10 million times. How do enzymes bring about such high rates of chemical conversions? The chemical which is converted into a product is called the substrate. Hence, enzyme that is proteins with three-dimensional structures included an active site convert a substrate into a product. The number of substrate molecules converted to products per unit by an enzyme molecule is called the turnover number. This exhibits the efficiency of an enzyme. All enzyme has a specific 3D structure as part of it known as the active site into which substrate fits. The point where substrate is bound on the active site is known as the substrate binding site. There is an obligated formation of an ER that is the enzyme substrate complex. This complex formation is a transient phenomena. During the state where substrate is bound to the enzyme active site, a new structure of substrate called the transition site structure is formed. Very soon the product is released from the active site. If P is at a lower level than S, the reaction is an exothermic reaction and is not supplied energy in order to form the product. However, whether it is an exothermic or spontaneous or an endothermic, the energy requiring reaction S has to go through a much higher energy state or transition state. The difference in average energy content of S from that of this transition state is called the activation energy. Enzymes eventually brings down this energy barrier, making the transition of S to P more easy. Nature of enzyme action. Each enzyme has a substrate binding site in its molecule to produce highly reactive enzyme substrate complex. This complex is short-lived and dissociate into its product and the unchanged enzyme with an intermediate formation of the enzyme product complex. The catalytic cycle of an enzyme action can be described as follows. First, the substrate binds to the active site of enzyme, fitting into the active site. The binding of the substrate induces the enzyme to alter its shape, fitting more tightly around the substrate. The active site of the enzyme, now in close proximity of the substrate, breaks the chemical bonds of the substrate and the new enzyme product complex is formed. The enzyme releases the product of the reaction and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. There are two use points by which enzymes are supposed to bring about chemical reaction. These are lock and key model and induced fit model. Factors affecting enzyme activity. The activity of an enzyme can be affected by a change in the condition which can alter the tertiary structure of the protein. Optimum temperature for enzyme activity is 30 to 40 in animals and 20 to 30 in plants. High temperature degenerates, denatures enzyme due to degradation of linkages in its polypeptide chain, whereas low temperature inactivates them due to reduction in speed of molecular movement. Most intercellular enzymes function near neutral pH with the exception of several digestive enzymes which work either in the acidic range of pH or alkaline. Low temperature preserves the enzyme in a temporarily inactive state, whereas high temperature destroys enzymatic activity because proteins are denatured by heat. With the increase in substrate concentration, the velocity of the enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity Vmax, which is not exceeded by any further rise in concentration of substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules, there are no free enzyme molecules to bond with the additional substrate molecules. We have a Michaelis-Menten equation. To determine the effect of substrate concentration in enzymatic reaction, Leudon, Michaelis, and the Maud Menten proposed a mathematical model and derived a relationship which mathematically expressed as 1913 Leonor Michaelis and Maud Menten. Is that V equals to Vmax, where a Km is a Michaelis mental constant that is a substrate concentration to produce half maximum velocity. Half maximum velocity, and V is the velocity of reaction. Vmax is maximum velocity, S is the substrate concentration. Then we have the inhibition of enzyme action. The activity of an enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme, and the binding of the chemical shuts off enzyme activity. The process is called the inhibition, and the chemical is called the inhibitor. Reversible inhibition is a temporary and is overcome by increased concentration of substrate, dilution, and the dialysis. Irreversible inhibition is of the permanent nature, in which the inhibitor combines with the specific functional group of enzyme through covalent bond. On the basis of competitiveness, enzyme inhibition can be divided into competitive inhibition and a non-competitive and allosteric inhibition. Competitive is reversible, which occurs due to the substrate or increased enzyme analog. Non-competitive is inhibitor forms a complex with the enzyme at a site other than the active site. Allosteric is the inhibitor binds to enzyme at specific site other than the active site and changes structure of active site to affect the structure bond binding. Allosteric modulation or feedback inhibition is an enzyme regulatory mechanism where a product or intermediates of a reaction functions as a temporary allosteric inhibitor, which combines with the regulatory site if its concentration crosses the threshold value. The end product inhibitor function as negative modulator, and the enzyme inactivated is called the allosteric enzyme. Michaelis Menten of the Km constant is not applicable in the allosteric enzymes. Inhibitor binds to enzyme at specific site other than the active site and change the structure of active sites to affect substrate bonding. Cofactors are called the non-protein constituents bound to the enzyme to make the enzyme catalytically active. In these instances, the protein portion of the enzyme is called the apoenzymes. Apoenzymes. Three kinds of cofactors may be identified, that is the prosthetic coenzymes and the metal. Prosthetic are the organic compounds and are distinguished from other cofactors in that they are tightly bound to the apozyme. For example, in peroxidase and catalyze, which catalyze the breakdown of hydrogen peroxide to water and oxygen. Heme is the prosthetic group and it is in part of the active site of the enzyme. Coenzymes are also organic compounds, but the association with the apoenzyme is only transient, usually occurring during the course of catalysis. Furthermore, coenzymes serve as cofactors in a number of different enzyme catalyzed reactions. The essential chemical components of many coenzymes are vitamins. Example: coenzyme nicotinamide adenine dinucleotide NAD, and the NADH can contain the vitamin niacin. Metal ions: a number of enzymes require metal ions for their activity, which form coordination bonds with the side chains at the active site and at the same time from one or more coordination bonds with the substrate. Example: zinc is a cofactor for the proteolytic enzyme carboxypeptidase. Metal ions, prosthetic group, prosthetic groups, coenzymes, the metal ions.